proteasome-mediated degradation, and how that's dependent upon a very unusual post-translational modification. And then more towards Tuesday, we'll begin getting into how modifications of histones can control gene expression. Okay, so last time I think we kind of finished up on hydroxylation. We were going through a set of different post-translational modifications to amino acids, and we need to know that lysine can become acetylated. You can see right here that as we do that, we take that terminal amino group, put that acetyl on there, and we convert this from a positive charge site to a neutral species. So this is really commonly done on histones. We talked about histones and their role in organizing DNA, the fact that a specified number of DNA bases wraps around those histone particles, and not surprisingly, that interaction between DNA and the histones is mediated by electrostatic interactions. The negative charge sites on the DNA backbone often interact with these positively charged groups on lysine. So as we introduce these sorts of changes, we are modifying the interaction between the histones, strength of the interaction between the histones and the DNA. Okay. Acetylation is carried out by enzymes called histone acetyl transferases. And these grab an acetyl group from a cofactor, acetyl-CoA. And when we acetylate the N-terminus of a protein, that tends to be irreversible, but these kind of side chain acetylations are reversible, and that reaction of deacetylation is carried out by another family of enzymes called the histone deacetylases. So forward direction, acetylation by histone acetyl transferases, deacetylation by histone deacetylase. Okay. Huge family of post-translational modifications are the addition of saccharide groups onto proteins. So these are introduced onto amino acid side chains, so they are a form of post-translational modifications. We talked quite a bit about glycoproteins when we were talking about saccharide structures, so I'm not going to go too much into this. I'll just tell you that a lot of these modifications, and we'll talk about several of them, these post-translational modifications happen at the endoplasmic reticulum as these proteins are being inserted into the lumen. So one of the things that you should realize is that the saccharide groups can be transferred onto a side chain oxygen, O-linked, or they can be transferred onto a side chain nitrogen species, and this is depicting N-linked glycoproteins. There's usually a family of enzymes that first sticks on a core set of saccharides, decorates them, and then sometimes trims them, and these oftentimes provide the addresses, the postal codes, to determine where a protein will end up being sent once it makes its way out of the endoplasmic reticulum. So I took a look at this structure of N-linked glycoproteins, and something doesn't seem right in there. I don't know if anybody sees it right away. But anyway, I'll leave it for you guys to find out what's wrong with that figure. Okay, so yeah, N-linked oligosaccharides attached to side chain nitrogens, as it is asparagine, and O-linked are conjugated onto the oxygen of serine or threonine. And then this just reminds you that these glycoproteins really do provide a key component of figuring out where proteins need to end up, whether they end up in a secretory granule or actually embedded in the plasma membrane. Okay, in addition to determining the location of a protein within the cell, post-translational modifications can also control activity. We were getting a little feeling for this in looking at phosphorylation, seeing how the addition of the phosphate group can really introduce a dramatic alteration of the electrostatic environment and have this massive alteration of the protein structure. Not surprisingly, when that's applied to an enzyme, these conformational changes can be so dramatic they can really switch an enzyme on and off. So some of these covalent modifications that alter enzyme activity for sure include phosphorylation, but also the addition of an adenyl group, a uridine, an ADP ribose, and then very often enzymes actually require the covalent addition of a group in order to carry out their enzymatic activity. And this can be the presence of a biotin group, or in the case of cytochrome C, these molecules need to have a heme cofactor inserted into the protein structure. Okay, so enzyme activity is often dependent upon post-translational modification. A lot of modifications can happen to cysteine. The thiol group, as we talked about before, is quite reactive. And when we were talking about protein folding and protein structure, we talked about the fact that the formation of cysteine disulfides very often can provide a staple to hold a protein structure together. And you can think about that as being a kind of post-translational modification. Oftentimes, proteins that are directed extracellularly include these disulfide cross-links. Because the extracellular environment is generally oxidizing, it promotes the formation of those disulfides. Some really unusual chemistry can happen to the cysteine thiol. The addition of a diatomic gas, nitrous oxide, can change this cysteine structure, and there's a really interesting biochemistry about this NO gas that's actually being a signaling molecule responsible for some interesting changes in physiology. Again, this manifests itself as a post-translational modification onto the cysteine side chain. All right, so kind of in the last step that we need to consider in the whole process of DNA going to RNA and RNA encoding protein. Okay, we make our protein, we modify it, and then we want it around for a certain period of time, and then we want to get rid of it, right? So the cell will control concentrations of key proteins, cycling them up and down, for example, at different stages in the cell cycle. And so this protein degradation is a really important issue. And the cell definitely controls how long a protein is kept around. Half-lives for proteins can vary from seconds to days. Actually, hemoglobin that's present in your red blood cells can stick around for a period of months. So what are the messages that exist within the cell and on a protein that tell the cell whether or not it should be degraded rapidly or slowly? There's something called the N-end rule, which is one really key aspect of protein lifetime. And if you take a look at the amino terminal residue of a protein, that gives you a lot of information on how long the cell wants it to be around. Now this is a little bit strange because we talked before about the fact that there's always one N-terminal residue on a protein structure as it comes off the ribosome, right? And what is that residue? N-terminal residue is first one that's synthesized, right? Synthesizing N to C. First one is methionine. Okay, great. So, like, what's the deal with the N-end rule? Why would there be any variation? It's because the N-terminal residue oftentimes gets processed and removed, just as we were talking about last time. So with that methionine present, half-life can be quite long, but other N-terminal residues can actually be quite destabilizing and signal the cell for a very short half-life and rapid turnover. So positively charged amino acids, for example, arginine and lysine, those might tell the cell, okay, I only want you around for half-life of just a couple of minutes. So what goes on then? The cell gets the signal, all right, this protein needs to be degraded quickly. How does that get manifest? What's the molecular mechanism that controls protein degradation? Well, this turns out to be an ADP-dependent process, and there's a whole machinery within the cell to control this. And it gets played out through a post-translational modification. This is a process called ubiquitination, and this is the signal for degradation. So ubiquitination is a process of post-translational modification where we're not adding just a few atoms. We're not adding like a phosphate group or a saccharide. We're actually adding an entire protein structure onto a side chain. There's a set of enzymes that are responsible for this conjugation of the protein. The protein is called ubiquitin. It is called ubiquitin because it is ubiquitous. You see this in many, many different organisms, and it's a highly conserved protein, 76 residues, very little variation in the sequence from one organism to another. So what we're going to do in this post-translational modification is that we are going to take ubiquitin, we're going to take the C-terminus, and we're going to add it onto a lysine side chain. And we can add one ubiquitin, or we can add several at that position, and depending upon the number of ubiquitin protein molecules on there, it will have different effects on the degradation. But once the ubiquitin is on the protein, it tells the cell, send it to the machinery for destruction and cleavage. And this is where that happens. This is the proteasome. It's a chamber of death. This is where proteins go to get cleaved up once the cell has no more use for them. It recycles. The cell is thrifty, doesn't want to synthesize amino acids and then just get rid of them. It may not need the protein, but it can sure reuse the building blocks. So this is the first step in that recycling process, taking the protein that's no longer needed and chopping it up into little bits. And here's the message that tells the cell to send a protein into the proteasome. Here's our ubiquitin post-translational modifications. This is the tag that says, take me to the proteasome and chew me up. We'll take a look at the structure in a little bit more detail. There's two regulatory entities on either side and a series of rings that constitute the catalytic core that have protease active sites within them. So here's one regulatory element, another regulatory element. So these are fairly symmetric. There's a bunch of beta subunits here that form a couple of rings that constitute what's called the core particle. This is where the protease active sites are. And I believe there's 6 different protease active sites that all ring around the interior of this chamber of death. The different protease active sites are responsible for carrying out degradation in different ways. So the proteins come in one side of this chamber, get chewed up by these proteolytic activities, and then short peptides are released that then go on to form individual amino acids that can be reused by the aminoacyl tRNA synthetases. It's a good idea to understand the role of the 3 types of enzymes that are responsible for providing this conjugation. There is the enzymes E1, E2, and the E3 ligase all play a role. E1 activates ubiquitin, and then E1 transfers that ubiquitin onto the E2 enzyme. E2 then forms a complex with the ligase. This is the enzymatic protein that actually is going to carry out catalysis of the post-translational modification onto a particular target protein. And this is a specific recognition event. This E3 ligase recognizes particular E2 enzymes in complex with ubiquitin and also recognizes particular target proteins, understanding that they need to be degraded, and then says, okay, I want to hit this type of target protein and conjugate ubiquitin that's on this particular E2 protein. So the E3 carries out its work. It attaches first one ubiquitin onto a lysine side chain, and then that can be the site for additional conjugation events, so we can attach multiple ubiquitin groups onto that target protein. And now we have our fully fleshed out signal. That polyubiquitin group telling it, okay, send me to the proteasome and chew me up. I want to stress that this is an ATP-dependent process, and we kind of take that for granted now, but it wasn't so obvious when this was first being studied. Yeah?
How does E3 recognize the target protein that needs to be degraded? Is there like a specific E3 for certain proteins?
Yes, there is. There is a huge number of different E3s, and the exact pattern of recognition, how a particular E3 ligase recognizes one set of proteins under one condition and not others, is ongoing field of study, and it's really interesting. And I'll get back to this in just a bit. But I want to stress that this is an ATP-dependent process. This was a surprise. Like, if you take a polypeptide and degrade it into its constituent amino acids, this is energetically favorable. It takes energy input to synthesize a polypeptide chain. And we've been seeing that on the ribosome. You know, that is a GTP-dependent process, right? And the aminoacyl tRNA synthetases also require ATP. It requires energy to build a polypeptide. So it was a bit of a surprise to find out that degradation is also ATP-dependent. How does that play out? Both with regard to the fact that the ubiquitin ligation is ATP-dependent, right? The E1, E2, E3 complex requires ATP to conjugate ubiquitin onto the target protein, and also the proteasome itself requires ATP to carry out its activity. I want to be specific about the nature of the post-translational modification. We're going to take a lysine side chain. Here's our primary amine. Here is the C-terminal position of the ubiquitin, and we're just going to make an amide bond. And so that's the reaction that's being catalyzed by the E3 ligase. And so the backbone of the target protein runs this way. Here is our side chain lysine, and then we're going to append onto it this big, huge protein structure. 76 amino acids, very stable fold, very well organized protein structure that goes on to a lysine side chain. That ubiquitin itself has lysine side chains. Well, that's interesting, because those lysine side chains can themselves be a substrate for the E3 ligase. So we can take a ubiquitin that's already on a target protein and ubiquitinate that to create ubiquitin dimers, trimers, tetramers, and to create a polyubiquitinated species. So this is an iterative process of post-translational modifications, adding one protein at a time and sending the signal to grade. These steps, again, for the E1, E2, and E3 ligase involve activation, ligation, and then polyubiquitination by the E3 ligase. And here's where I want to stress this issue of specificity. There's many of these E3 ligases. There's several of these E2 enzymes, which associate with the E3 ligase. And there's only a couple of these E1 enzymes that are responsible for activating ubiquitin and transferring it to E2. So ubiquitination of proteins is targeted and controlled. And it's controlled in the sense that the E3 ligase, there's hundreds of these things. They are going to be responsive to particular sets of target proteins, and they will understand and know when those proteins need to be degraded. So the recognition of the proper substrates is by this huge family, hundreds of E3 ligases. All right. So that was a pretty interesting post-translational modification. This was thought of being like a total oddball, like we're not adding a saccharide, we're adding a whole protein, but now we're beginning to appreciate that there's actually a whole set of different protein modifications that can be introduced. And I don't want to really spend too much time on this, but just as there's ubiquitination, there's also something called sumoylation that takes this compact protein, the sumo protein, and also conjugates it onto a protein side chain. We now know there's, in addition to ubiquitin and sumo, there's also hub1, fat10, NED, and so you can find biochemists who are really interested in NEDylation. You know, it's getting pretty arcane, and again, don't want to spend too much time getting into the details of these. The take-home message here is that post-translational modifications include proteins. The most common, ubiquitous one is ubiquitin, but there are other proteins as well. Control of protein concentration and control of degradation turns out to be really important as we think about going from one stage in the cell cycle to another. And this is largely controlled by a family of proteins called cyclins that tell the cell to go from one phase to the other. And so being able to have increases and decreases in a very timed way in these cyclin proteins is really crucial in things like governing exit from mitosis and progressing into the next cell cycle. And these cyclins, what determines whether or not they're going to be upregulated or degraded is largely reliant upon ubiquitin-dependent proteolysis. So a lot of interest in finding out how particular E3 ligases target cyclins at very particular times to control cell growth and division. Yes? How does ubiquitin get degraded? I don't know that it really does. It may not be through a proteasomal-mediated process. In addition to degradation in the proteasome, proteins can also get degraded in lysosomes. So it may be through that less specific process that ubiquitins are degraded. So what to know about post-translational modifications? You need to know that there's N-terminal processing, proteolytic cleavage, phosphorylation, lipidation, these smaller covalent modifications to protein side chains such as carboxylation, methylation, hydroxylation, and N-acetylation. Glycosylation is a whole, field unto itself. Cysteines can be modified, including the formation of disulfides, and then ubiquitination, controlling degradation. So that's the overview there for post-translational modification. So now I'm going to switch gears and get into control of gene expression. But before I do, any questions about post-translational modification. All right. So maybe we could just spend a little time thinking about what might the cell do to control protein concentration. Yeah?
Sorry, I do have a question about post-translational modification. You said that the E3s receive a signal for which is the target protein. Where's that signal coming from?
Right. In some cases, it can be phosphorylation. So the E3 ligases will look for a protein that's been phosphorylated, and that will be the signal to them that, okay, now I can recognize you and target you with ubiquitin for degradation. There are other alterations, and E3 ligases are responsive to other events within the cell to turn on degradation of their target protein.
And in terms of cyclins, are those like mostly cyclical, or are they some cyclical and some not, or is that like how is the cyclical part of that involved? I guess.
I'm not sure what you mean by cyclical.
Like the phosphorylations and other types of signals, are those regulated like on a cyclical basis in terms of cellular processes? Like one leads to the next, so it repeats, or? Yeah, right.
As the cell goes through the cell cycle, that those signals to either synthesize them or degrade them, they cycle up and down as the cell repeatedly goes through the different stages of the cell cycle. Yeah. Okay, so let's think about how the cell might might be able to control levels of proteins. Okay, ubiquitination and proteasome-mediated degradation, we got that down. What else could the cell do to control protein levels? Yeah. Right. So we could alter the expression of a certain gene and control whether or not an mRNA is going to be generated. Okay, so that would relate to control of mRNA synthesis. What else could the cell do? Yeah? Okay, right. So the mRNA itself could be degraded, and we'll also talk about ways that mRNAs can be complexed to keep them silenced. Right? So definitely at the level of transcription, there's a lot that can happen. So we can control the generation of a particular primary transcript. We can also control post-translational processing, right? We talked quite a bit about what is required to generate a mature mRNA. So we can influence the extent to which we generate that species from the primary transcript. Control of mRNA lifetime is another mechanism. And then as far as controlling concentrations of protein, well, we did talk about the fact that post-translational modifications can determine whether or not a protein is active or inactive. And so that's actually a way of controlling the concentration of an active species for an enzyme. And then protein degradation. Targeting a protein to certain organelles within the cell maybe would sequester a receptor within the cell so that it can be released onto the protein surface at some other point in time is another way to control the concentration of protein in its active state. But let's really focus now on gene regulation, whether or not an mRNA is going to be generated. Certain genes are just basically on all the time, and we call that constitutive gene expression. And others, the cell only wants at particular points in the cell cycle or in response to a particular signal or a particular stress. And so those are, that's termed regulated gene expression. And the cell can either turn things up or turn things down. When additional expression is required, that's called induction. And when the cell turns down expression of an mRNA, that's called repression. we talked before when we were discussing generation of an mRNA, the promoter region that determines the interaction of polymerases at the start site for an mRNA. And proteins can bind right around those promoter regions to alter the extent to which transcription occurs. And we've already taken a pretty careful look at one of these proteins, and those are the specificity factors. We call those sigma factors. Right? Those were complexing directly upstream of the mRNA start site and recruiting RNA polymerases at that start site. So those sigma factors are constituents of RNA polymerases that actually get the process going. Repressors are proteins that can bind in that promoter and physically block the polymerases from carrying out their work. Activators bind in the neighborhood and recruit the polymerases to the promoter region. So these play opposing roles. And then these sites where the repressors can bind, those are called operators. And those are specific sequences on the DNA. There are specific protein-DNA interactions at those positions, and we'll take a look at a couple of them today. So what determines whether or not a repressor or an activator will actually be capable of binding in a particular promoter? Very often that's determined by what's called an effector molecule. And this is the signaling entity that tells those repressors, okay, now you should engage with your target DNA. Oftentimes these promoters control the expression of a set of genes, and those clusters of genes that are all regulated together are called operons. And we'll take a look at a couple of operons today, the lac operon and the trip operon, some of which may be familiar to you guys. Dealt with these before. Okay, so specificity factors, once again, I just want to remind you that upstream of the RNA start site, we have these regions of DNA, and a component of the RNA polymerase machinery, in E. coli we call that the sigma subunit, recognizes those DNA sequences and brings the polymerase to that site to initiate mRNA synthesis. So if we want to all of a sudden express a set of genes in response to some signal, for example, in response to some kind of heat shock, well, what the cell can do is just swap out the presence of the sigma subunit, and then all of a sudden a completely new set of genes that have a different operator region will then end up getting expressed. There are some interesting mechanisms by which small molecule ligands can influence the activity of activator proteins or repressor proteins and determine whether or not they're going to be carrying out their work. And so there's 4 different modes of regulation here. This gives you the overview for how ligands can interact with repressors or activators and turn transcription on or off. And I'll go through each one of these here. There is ligand-regulated repression. Okay? So this is a case in which we have a repressor molecule binding into an operator, and that repressor molecule will bind a small molecule effector, and in the presence of that bound effector will sit on the operator and prevent the mRNA from being generated downstream. But sometimes small effector molecules actually will liberate the repressor off the promoter, and so we call that ligand-regulated de-repression. So the repressor will normally bind to the operator without the presence of the small molecule. That effector binds, and then the repressor is released, and mRNA can be generated. In addition to repressors, we have activators. Okay, so we're showing these here in green. And again, small molecule effectors can either encourage or discourage these activator proteins from carrying out their job in binding upstream of the operator. In this case, the effector regulates activation. The activator wants the ligand bound to promote binding of the polymerase, and in this case, binding of the small molecule bumps the activator off of the promoter area, and only in the ligand-free state will the activator recruit the RNA polymerase. In this case, it's called ligand-regulated deactivation. So the promoter and the associated binding of a repressor or an activator in this regulatory region can control more than one gene at a time. We have a bunch of genes downstream of a particular promoter site. They can all be transcribed as one unit, and we call those operons. And we're going to take a look at how operons are regulated in prokaryotic systems. And we'll begin by taking a look at the lac operon. So this is some genes that control whether or not lactose can be readily used as a source of metabolites for the cell. The cell needs to be able to recognize when lactose is present at high concentrations and when the cell wants to begin using that as an energy source. There is a transporter protein within the cell membrane, galactoside permease, that allows lactose to come into the cell, and then a couple of enzymes that exist within the cell to transform that lactose into the constituent monosaccharides, galactose and glucose. And so the cell's not going to really need these enzymes around unless the cell is prepared to use lactose as a carbon source. So this needs to come under regulatory control. So both of these enzymes need to be controlled simultaneously, and so they're put under the control of one promoter to constitute an operon. So let's think about how we might want this to work. Here's our promoter region right here, P. Here is an operator, so this is where a repressor protein can sit down. So when that lac repressor is bound right here, the polymerase cannot move through and generate these mRNA for these enzymes, galactosidase and galactoside permease, that are responsible for using lactose as a carbon source. So in this case, one of the steps in the conversion of lactose to the monosaccharides is the formation of allolactose. So this can be a signal to the cell like, hey, we've got plenty of lactose around. You might want to think about using this. So if I tell you that this lac repressor binds allolactose, then how do you think this system will respond? What do you think that allolactose will do to the lac repressor and influencing the operation of this set of genes? Yeah.
Ligand-regulated derepression.
Okay. So binding of the allolactose results in a conformational change into the repressor that pops the repressor off. It disassociates that repressor. And yeah, it allows all these lac operon genes to be expressed. So this is a case where the repressor is bound in the absence of the small molecule effector, and the allolactose then liberates that repressor and allows these genes to be expressed. The engagement of the LAC repressor with its target DNA is something I want to pay attention to, because this is kind of like the molecular level of how gene expression gets carried out. How actually do these repressor proteins know where to bind, and how do they respond to the presence of these effectors? So we can draw the DNA like this in forming a loop when it is bound by the repressor molecules. And you can see that this would be a major change in the DNA structure that could have a huge influence on whether or not RNA polymerase is going to be able to sit down properly and begin cranking out the corresponding mRNA downstream of this site. There are two operator regions upstream of the LAC operon, and so the LAC repressor binds at a couple of them and forms this homotetramer that encourages this loop-type formation to form by DNA. So here are the LAC repressor proteins, and you can see that they've kind of reached out and they're binding onto DNA. And one of the really interesting questions is, how does this protein know to bind at one site versus another? We've seen plenty of examples of proteins capable of binding DNA largely through electrostatic interactions, but these tend to be not specific. If we don't get this right, these repressors are going to bind indiscriminately, and they'll have an influence within the genome where they're not wanted. We've been able to study exactly the influence of small molecule effectors on the structure of the LAC repressor. So we have X-ray crystal structures of the LAC repressor in the presence and absence of this molecule, IPTG. So does that ring a bell to anybody? It does to you. Why? You probably used it, I bet, right? Has anybody used IPTG? I'd be surprised if you guys haven't. Okay, so when were you using IPTG?
Like as part of a synthetic biology thing where you use like an IPTG inducible promoter to drive expression of a certain gene.
Exactly, an IPTG inducible promoter, right? So when students in my lab want to do recombinant expression of a protein in a bacterial host system, they put a plasmid for the gene encoding their protein of interest into bacteria, they grow up the bacteria, and then like, okay, now I want the E. coli to crank out my protein. They toss IPTG into the culture medium. That's kind of like magic. That's just like what the protocol tells you to do. Induce with IPTG. What's going on? Those genes on that plasmid are under the control of this LAC operon or something very similar to it. And it is the binding of this isopropyl thiogalactoside, IPTG, into that repressor that kicks the repressor off, and now the bacteria are off to the races, cranking out your protein of interest. Look at the changes in the structure. It's really dramatic. This small molecule binding into the repressor has these huge influences on the structure of the repressor that alters its ability to bind into DNA. Sequence-specific recognition is really crucial, right? These repressors need to bind to that promoter and no others. It needs to get it right. So how do we take a look at a region of DNA and figure out, okay, this is the right sequence? And this gets back to something that really troubled Watson and Crick and Pauling and others who are thinking about functional DNA molecules. The fact that the sequences that determine one region of DNA for the other in a DNA double helix, they're tucked away in the inside of that B-form DNA structure. So how do we read a sequence? Well, although the base pairs are on the interior, they actually can be accessed to a certain degree, both through the minor groove and the major groove, right? And so when we're taking a look at bDNA structure, we're taking a look at the ridges formed by the backbone of that bDNA, and we saw that there were kind of valleys within the cylinder formed by the double helical DNA. And those valleys, there's two of them. One is wider, one is narrower. Those are the major grooves and the minor grooves. And you can kind of get a feeling for a particular pattern of chemical functional groups for each one of these base pairs present in the major groove or minor groove that a DNA binding protein could recognize. So this is what a interaction of a protein side chain with, let's say, a TA base pair or a CG base pair might look like. Typically, these are mediated by hydrogen bonds. Charged residues can also be important. So kinds of amino acid side chains that are playing a role here, ones you see over and over again, are asparagine, glutamine, glutamic acid, lysine, and arginine. And it would be really great if we could find some kind of code for complementarity, right? So we know what the Watson-Crick rules are for recognizing a particular DNA sequence. Line up the proper hydrogen bond partners. It would be nice to have a one-to-one correspondence here as well. But there isn't any simple code. We can't take a look at, like, say, okay, CG, CG, AT, AT, TA. Like, okay, for that particular sequence, I just need to line up arginine, lysine, asparagine, asparagine, glutamine, and it'll recognize that sequence. There's no one-to-one correspondence. So this code has been really difficult to crack. But how might you take a stab? Does anybody have any suggestions about trying to understand why a particular protein sequence might be able to recognize, in a general way, a particular DNA sequence? Any ideas? Let's say we've run out of good ways to do experiments. Can we still study this? Are there ways to study biological molecules in the absence of actually working with them at the bench? Yeah. Yeah. So computer modeling, as I told you before, has been really helpful in predicting protein structures and understanding folding properties. And we're beginning to learn how to use computational techniques to design potentials or equations that will predict what type of amino acid sequences will be capable of recognizing particular nucleic acid sequences. Very, very tough problem, but making a little bit of headway. All right, so let's take a look at the interface between a protein structure and DNA in the context of this lac repressor. And what we need is we need to grab a bit of the protein structure, position it right next to the DNA, and have it be really well ordered and positioned so that it can reach its side chains into the major groove or minor groove. So here's our lac repressor dimer. And here are these little bits of the protein structure that are really nicely, compactly organized to position like some of these loops that are attached to these helices to reach in and engage with the base pairs in the interior of the DNA. So each of these dimers binds to an operator sequence. It's about 22 base pairs long, and it binds with pretty good efficiency. The dissociation constant is about 10 to the minus 10 molar for the KD. That's pretty good. And you should think about, like, okay, well, how specific would that allow you to be? And if it was dissociation constant of, let's say, 10 to the minus 4 molar, you'd be like, okay, well, it'll bind that sequence, But maybe there's a whole bunch of other sequences in the entire genome that it can bind as well. This value, 10 to the 10 to the minus 10 molar per kb, has been calculated to give a really good discrimination ability. About 10 to the 6-fold tighter binding to its target sequence versus other random sequences within the DNA. And that's crucial, because once again, it needs to bind to those 22 base pairs, not any of the other 4.7 million base pairs that might exist within a bacterial genome. That specificity is crucial. Okay. Here's where the lac repressor binds. Here's the RNA start site. And here's the promoter region, right? So this is where that sigma factor might bind. And you can see that when that lac repressor is sitting in that operator, that RNA polymerase is blocked, it's occluded, and we're not going to be able to generate an mRNA message when that lac repressor is present. There's really nice complementarity between the lac repressor and the DNA. Okay, so the DNA is shown here in blue, the lac repressor in gray. And you can look at a co-crystal structure, pull the two components apart, and ask, where is their complementarity? Well, hydrophobic patches on the surfaces of these biomolecules are lined up very nicely, and then positions where there's hydrogen bond interactions between the protein and the DNA are shown within this major groove there in red. So that explains some of the features allowing for specificity in protein-DNA interaction. It's actually also kind of an interesting problem in protein structure, how to get the protein to fold up in such a way that it can orient a very compact part of its structure and reach into the DNA. Different DNA binding proteins solve this in different ways. Oftentimes there's some kind of component that really helps to define the structure. And so for these zinc finger motifs, you'll see that here, this portion of the protein structure that's associated with the DNA is complexing zinc right here that's defining that structure. 4 amino acids are in complex with the zinc. Anybody look at that and figure out what that motif is, what 4 amino acids are there? These two. You recognize what those are? These are aromatic, but that's like a nitrogen-nitrogen here, so that's not a phenylalanine. So these are 2 histidines, and then these yellows indicate sulfur atoms. So this is a very classic motif within these zinc fingers, dicis, dihis. 2 cysteines, 2 histidines that bind zinc. So binding of these, it needs to be tight. The binding of any individual module is not quite so strong, so very often you see these polyvalent interactions where you see dimers and tetramers recognizing their promoter sequences. Here's an example where a dimeric protein is recognizing a particular DNA sequence. These are leucine zipper motifs, so long extended alpha helices. They complex on either side of the DNA, and they are extending part of this protein chain to recognize particular base pairs. This is the DNA contacting portion of these leucine zippers, and here is the zipper region. And we have leucine at every 7th position, shown here in red. Why 7? Why every 7th residue is going to be leucine? Yeah?
There's 7 terms. There's 7, amino acids per term.
So it does have to do with the helical repeat. there's 7 amino acids for every 2 turns of the helix, right? Here's one of our leucines, 1, 2, another leucine, 1, 2 turns, another leucine. So positioning them at every 7th residue lines them up really nicely so these leucines can interdigitate with each other and form a very nice dimeric complex. That stabilizes the alpha helices and positions them really nicely to recognize their particular DNA sequence. And then in here, we have got a bunch of arginines and lysines in this DNA binding region. No surprise there. Positive charge sites to provide charge complementarity for the DNA backbone. And so these are the sequences that you might see for some typical DNA binding regulatory proteins. Some of these may be familiar to you. John Foss, GCN4. Again, the leucine zipper sequence is here. You see those leucines every 7th position. Positively charged amino acids here, and then DNA binding region within this portion of these leucine zippers. All right. I'm just going to give you a brief introduction to the trip operon. I want you to be somewhat familiar with this. When we get into the nitty-gritty of how operons work, I'll probably be keyed in on the lac operon, but this is a kind of a cool variation. This is a set of genes that control whether or not tryptophan is going to be expressed within the cell. And obviously that's something that needs to be regulated. If the cell already has plenty of tryptophan or it's not going to be rapidly dividing, don't make the tryptophan if you're not going to use it. So the expression of these particular genes, all within an operon, is going to be controlled by this operator. And there's 2 ways that the expression of these genes within the operon are controlled. One is really very similar to what we've just been talking about. There is a repressor, right? And the repressor is responsive to a small molecule effector. And what better effector to use to determine whether or not you want to make more tryptophan? You want that repressor to be responsive to tryptophan itself. Sure enough, in the presence of tryptophan, that repressor is going to bind at the operator and shut down the expression of all these genes. Makes total sense. Have your repressor bind the end point, the end molecule of this pathway, and shut it down. We don't need these genes to make more of this amino acid. In addition to that, the expression of these genes is also controlled by an attenuator. I'll get to that in just a second. The interaction of the trip repressor with DNA is very similar to what I've been showing you before for DNA binding proteins. It does bind as a dimer. Here you see the tryptophan present in the repressor and determining that, yes, the repressor should be in the DNA binding form. But here's this really cool attenuation process that is a second mechanism for determining whether or not this set of genes is going to be generated. Let's take another look at this region, this operon. Here are all the enzymes, the genes for all the enzymes that are going to be used to, for example, make the tryptophan synthase. But right at the start here is what's called a leader sequence. That's not giving rise to any functional protein in this system. This is the mRNA that has this leader peptide sequence in there. And now here is the portion of this mRNA that's being transcribed that's going to actually begin making a functional enzyme. So it's really interesting to see, this seems to be like a really short, useless little peptide. We've got the start AUG here, you've got a stop UGA here. It's just a short little peptide, and what makes this really unusual is there's 2 tryptophan residues, 2 tryptophan codons right here, right next to each other. What is that doing? Don't forget that in a prokaryotic system, the polymerase can be making the mRNA while the ribosome is beginning to work on that nascent mRNA and already beginning to crank out a polypeptide product. And that's what's happening here with this attenuator region. So the polymerase is making mRNA, the ribosome is reading it, and then if the tryptophan levels are high, no problem. It goes right through and goes WW, makes that polypeptide and moves right down the mRNA. As it does that, the ribosome engages this region, this number 2 region here, and it allows this portion of the mRNA that's been generated by the RNA polymerase to make this hairpin structure, which tells the RNA polymerase, okay, that's a stop signal right here. You need to terminate RNA polymerization. So the RNA polymerase leaves, and all of those downstream genes never get made. Now let's imagine a situation in which the cell needs tryptophan. That ribosome is going to hit that same sequence. It's going to see the codons for tryptophan. There's not going to be any tryptophan around. So it's going to sit there and wait. Waiting for EFTU. Please bring me tRNA with tryptophan on it. I'm not seeing anything. I'm just going to wait here until I find the tRNAs I need. The RNA is going to continue to be generated by the polymerase, and that's going to give this region 2 an opportunity to complex with region 3. This attenuator hairpin structure will never form. The RNA polymerase will never see that termination signal, and it will continue to march through and make all the mRNAs that are in that operon, allowing the synthesis of more tryptophan. So that's the way that trip operon works. Okay, questions about the lac operon, trip operon, before I wrap it up? Next time, we'll talk about gene regulation in eukaryotes. We'll talk about modification of histones by acetyltransferases and how that can play a role in activation. We'll talk about the fact that in eukaryotes, we really oftentimes will rely upon positive regulatory mechanisms. So in bacteria, we're often talking about repressors and de-repression. We don't really need repressors in eukaryotes. So you might want to think about why that's the case for eukaryotes. We don't need repressors. Regulatory proteins and protein complexes tend to be larger, more complex in eukaryotes, and transcription and translation are physically separated events, and so that has an impact on regulation as well. I want to show you this video. It's kind of an amusing way to think about getting prepared for your midterm, but I'm going to include this summary slide here. It'll be available on the lecture notes. Definitely know how protein concentration is controlled through different processes through both transcription and translation and degradation. Understand the roles of repressors and activators. Know the 4 different ways effective ligands can control gene expression, and then have some familiarity with the trip operon, but certainly with the lac operon, have a good understanding of how that works. Okay, so at this point, I'm going to show an amusing video about protein synthesis. For those of you who are eager to get to dinner or to get to studying, this is not required, but I just get
Was brillig and the slithy 30S ribosome did gyre and gimble in the wave. 30S ribosome.
This is an interpretive dance from UC San Diego in the 1970s.
Paul Mimsy was mRNA, that colored message unit array.
So they're wearing colored balloons on their head to represent the different bases.
mRNA.
30S is here, waiting for the mRNA complex. 9 Delgarno sequence. Initiation factor.
Found they were in the glorious sun by initiator factor 1.
So now the mRNA is sitting on the 30S.
Initiator factor 2 went searching for tRNA, who bore the plasmid amino acid.
Okay, so this is going to be our initiator tRNA. Boom! Hydrolyzed. Yes.
Then, as the uppish bonds were formed, the 50S ribosome untamed, came whiffling through the tallgy grass and burbled as it came.
I had to live through this. I'm allergic to hippies. So this is the
T
FTU.
factor plus GTP gathered in tRNA.
Second tRNA.